Okay, in this talk, I'm going to, the name of this talk is very ambitious because I am very interested in evolutionary biology and evolutionary phenomena in general. And obviously, this talk is supported by different institutions that I want to thank. DICTA is a foundation of where I am working as a general coordinator with Tomás. Tomás is the director of DICTA, is in Chile principally. The University of Chile is the university where I am doing my PhD. Obviously, I need to thank also to the government of Chile for my scholarship, and obviously for this university, the BUB, and the CLEA, the Center of Leo Apostel, that you invite me to have this talk. So, and the other thing that is important, obviously I put my email here if you are interested to talk with me, or maybe get deep in some aspect, you can write me, okay? Okay, so obviously this, as Tomás said before, this is a part of this seminar is possible because it's a part of this big project, Origin of Culture, that is possible because it's funded by John Templeton Foundation. And, okay.
No, it's okay.
Okay. And in this talk, I'm going to speak. I'm going to develop in general 4 points. This talk will be divided in these 4 topics. First of all, I will speak about evolution of life and their processes very briefly. A wide topic of evolutionary processes. In the second part, I'm going to introduce a little bit about reaction networks and chemical organization theory. In the third part, I'm going to speak a little bit also of works that I have been developed with Tomás about the applications of these reaction networks, but in endosymbiotic systems. And finally, I'm going to speak a little bit, I'm going to introduce, because this is a topic that I like so much and one of my interesting, that is reaction networks applied to Evo. Okay, so when we speak about evolution, obviously I think that in general, all of us know a little bit about evolution. And a little bit about evolution of lives. In the case of evolution of lives, when we speak about evolution, we refer in general, we have in mind in general this kind of trees, okay? For example, in this case, we can see in both trees, we can see different groups that split from an ancestral group, okay? So evolution is the change of different things that are related to life. For example, the change of organisms that are related to molecular change, for example, DNA or proteins, change of the morphology, for example, and maybe I can admit the people when they are coming. Yes, and another important thing about evolution is that we relate this evolutionary process to the timing. For example, here in the up of the picture, we can see the different periods of the tree, of the history of tree, that obviously in this kind is related to millions of years, but evolution can occur in thousands of years, and obviously it's also occurring now. We cannot see that because we cannot see the results of evolution in this moment, but it's something that is always happening. And it's also related to different events of the Earth in this case. And when we're thinking about evolution, as I said before, we need to focus in the similarity and in the difference of organisms related to their, for example, in different levels, in the levels of molecules, for example, in their development, in their morphology, in their plasticity, with a lot of levels, okay? Okay, maybe I'm going to. Okay. Okay. So when we think about the evolutionary processes, okay, we are talking about a lot of processes that are related to evolution, okay? Just only for example, when we focus in this more current theory of evolution that is in general is discussed today, is discussed currently, the extended evolutionary synthesis consider not only the organism that, for example, one species that is changing for a time. It's not only important the organism for this view of evolution, but also their interaction with the environment, okay? So for this current view, because previously view of evolution, for example, Darwinian selection or the modern synthesis, in generally was focused in the organism or in the population of organisms of the same species, okay? But in this more current view, it's more important the systemic view. So in this case, for example, the systemic view involves the interactions between the organism to the environment and from the environment to the organism. For example, in this case, they show the natural selection and imposition from the environment to the organism, the developmental plasticity also, the developmental bias from the organism to the environment, and niche construction, okay? That is some of the process that this picture show. But I want to show with this picture, with this slide, this systemic view of evolution. I want to emphasize of this systemic view, because I think that in this theory there are two important things, okay, that I think that are implicitly, okay, that are implicit in this view. One is the self-perpetuation of the organism itself, and the other thing is the interaction, the constant interaction between the organism and the environment, and their constant change, because this relationship is also dynamic. The organism, obviously, for example, with these two processes, can change the environment by their behavior, for example, and then can change together, making a history in common, okay? So I think that is one of the important things that we need to remember for the rest of the presentation. And also I want to emphasize only to have in mind that the evolutionary process that contribute to the inheritance, and obviously when we think about inheritance, reproduction, that is the result. The phenotypic evolution is a consequence, obviously, of the inheritance of the organism, okay, in their population. And this view show that there are a lot of processes that are involved in this inheritance, in this phenotypic evolution, and in different levels. Okay, for example, in this case, in processes that generate novel variation as genetic change, gene expression, developmental processes, environmental change, and there are another level that are processes that via selection, for example, developmental bias or niche construction, and another processes that modify the frequency of heritable variation. Okay, so when we think about evolution, obviously we are we have to have in mind also the multi-level evolution. If I want to say in other words, the evolution of one of those levels. Okay, and in general, there are also obviously in general this very wide view of evolution have been developed, have been changed in a in their history from the beginning, from the Darwinian view of evolution, like natural selection that is in yellow here, have been changed to the modern synthesis that is in green, and in general we are now in this part, in the blue one, that is the extended the extended evolutionary view that involves obviously different disciplines. Okay, so one important thing also is that we know that this is necessary to have a multi-level view of the evolutionary phenomena, and also because the disciplines here that obviously have things in common, but one thing that in general happen is that you can focus only in one discipline, and maybe because it's difficult to have a very wide view of evolution, you just only use the tools and the concept and the different ways of study your research, but only focus in one of these fields. Okay, so this I put this picture because give us a better wide view of evolution also. Okay, and this is one, this wider view of evolution, this multi-level view, is one of the things that we need to have in mind. And after this presentation, finally at this presentation, I'm going to return to this topic. Okay, but now I'm going to speak a little bit about other things. Sorry. Okay, because one interesting thing obviously is, well, so which tools, or maybe what is necessary, or how we can approach to the evolutionary process by a systemic view. Okay. So in this talk, we wanted to present you. I don't know if the people can look the, no?
They see the full.
The full picture. Okay. Yeah. Okay. So we want to present here the reaction networks and the chemical organization theory. Okay. What are reaction networks? We show here first here a list of reaction, and in B here a graphical representation of a reaction networks. A reaction networks, in their more basic aspect, is a group, a set of reactions, and relate a set of species but are related also with a set of reactions, okay? So this list of reaction here is very, very small, but could be very big also. And what is important to know is that the species, for example, we have here A, the species A, C, B, okay? And we describe their interaction with like something similar to a chemical reaction, okay? And also, in this case, we cannot see here, but we will see after in a couple of slides more that in their reaction it's possible also to put parameters to show to describe how is the rate of that reaction compared to another reaction in the same system. Okay, so for example, in this list of reaction, we have in the reaction 1 an species A, E, that is describing a self-reproduction. Okay, in the case, for example, in the reaction 3, we have the species B plus C that generate B plus 2C. Okay, in this case, we can describe a self-reproduction of the species C, but mediated or catalyzed by the species B. In the case, for example, of the reaction 2, we can see that there is a species E, A, that plus C, but only is generated the species C again, okay? Because in that reaction, for example, this reaction show that A is destroying, okay, to generate, to perpetuate C. We have here the graphical representation, but one of the other things that are very interesting related to the reaction networks is that this list of reaction could be very big, and could be organized in different group of reaction. For example, in this picture, we can see that there is an organizational structure. Okay, so we looked the different species, but that are grouped together depending of their reaction, depending of their kind of interactions in different subgroups like modules. Okay, but this is something that I want to emphasize after, because there is another picture that I think is better. This one is because when we have a lot of reactions, okay, one of the more ambitious, I think, and one of the things that I am very interested is to generate this abstraction of the phase space. What is this? Is all the reaction network. When you have all the reaction, you can put you can understand this general picture. You can describe based in their interaction, you can describe this whole picture, finding different states of these subgroups. Like this picture put here, we can find, for example, in black, subgroups that not have any properties, but we can find also closed systems. We can also find self-maintaining systems that are in semi-organization, and one of the more ambitious goals of this approach is to find the organizations. What is organization? Is an organization is a self-maintaining and a closed system that is a good candidate to achieve a stationary state. Okay, so this is very important, because when we have a very big view with a lot of list of different reaction, different species, when you find this kind of organizations for an evolutionary point of view is very interesting, because you can maybe relate this stationary state to one kind of traits, for example, that is more generally that doesn't have so much change in the evolutionary process, for example. And this could be related also because sometimes those kind of traits maybe are more pleiotropic, or maybe they have more relation with other species. Okay, so that is one of the things that I'm going to speak a little bit after. Okay, no. Sorry. Okay. So one of the advantages of the reaction network approach is that we can see some of their advantages in this table, because obviously we know that there are others kind of approach, for example, dynamic equation or differential equations, networks, agent-based models, for example, and but in the case of reaction networks plus plus chemical organization theory, sorry, in the case, for example, if we can see there in the case of differential equations, in general, it is possible to use a few species. In the case of networks and agent-based, many. In the case of interaction, we can describe in general a few. In the case of equations and networks, for example, in the case of mechanisms, it is possible to describe mechanism in the case of differential equations, but in the case of network is difficult. It's not possible to do that. In the case of the agent-based is partial, and the other thing is the analytical tools, because the analytic tools in the case, for example, of differential equations are rich. In the case of network also. In the case of agent-based modeling is poor, but when we see the reaction network approach or the chemical organization theory, we can find that in the case of a species, it's possible to use many species, it's possible to use many interactions. In the case of the evolution dynamic is indirect. In the case of mechanisms, it's possible to put some mechanism, to describe some mechanism in the model, and also it's possible to use because there are so many analytical tools. Okay, it's very rich. So that is one of the advantages of the reaction network. And another advantages that I describe is that, no, I don't describe, I emphasize, is that, for example, here we can look that obviously we can choose different, totally different processors or concepts that we can model using reaction networks. And also we can use conceptual processes that are more related with abstract things, but we can use this concept in the same model. That is not very common. That is not possible, for example, in the networks-based models. Okay, but in this case, is also we can use concept, we can use abstract thing at the same model with, for example, I don't know, genes or maybe molecules or other things that we want to express. And the other thing is that is related to the processes, because when we or for example, in the models that we have developed with Thomas, we wanted to describe the system with all the processes. We describe the processes related to their constant parameters. Okay, we can speak more about that after. And other thing, well, I wanted to show you as an example that is possible also with this kind of reaction, describe this kind of interaction. For example, in this case, different interaction from the ecological interactions, okay? Okay, so after this, we want to speak. I want to speak a little bit about our system, our approach that we have developed using this this kind of models. And first of all, because we focused in endosymbiosis. I'm going to speak a little bit about endosymbiosis because we choose why we choose this kind of system to as one of the first approach to the evolutionary process. Why is important? Endosymbiosis refers to the coexistence of living matter where one organism lives within the body or within the cells of another organism. In general, the endosymbiotic organism is one cell. And generally, this relationship benefits both of the organism, unlike the parasitic relationship where one feeds off the other. Okay, in general, endosymbiosis is a plus relationship interaction because in general it's related to a mutualism interaction that is benefits to both of the organisms involved in the interaction. And endosymbiosis can be understood, can be studied for a lot point of view, obviously, but there are two that are very interesting in the field of evolution and ecology. One is their evolution, for example, the coevolution of the species, because, for example, there are some groups that have an endosymbiosis relationship and perpetuate this relationship from millions of years. Okay, we want to discuss after a little bit more of that. And the other important aspect is the interaction itself. For example, in the first aspect, we can think of thousands of millions of years. But in this interaction, we can look the interaction or how the both organisms benefits each other in the moment, okay, at the moment, or I don't know, in the present, for example. So these two approach. For example, in the case of the evolutionary viewpoint of endosymbiosis, why endosymbiosis is interesting, why endosymbiosis is famous now, is because maybe you know that this woman is Lynn Margulis, one of the women that took this theory of endosymbiosis that begins at the beginning of the 20th century, and she developed this theory. She with an empirical viewpoint that she did a lot of experiments related specifically with bacteria, because we know here that bacteria that mitochondria and plastids. Mitochondria is these organelles that in the eukaryotic cell that gives to the eukaryotic cells energy, okay, ATP, for example. And the plastids are the typical organelles that in plants gives them the possibility, the chance to make photosynthesis, okay? So, but this kind, in especially chloroplast. So, this picture show that from bacteria, because bacteria have a lot of things in common with mitochondria and plastids, and obviously Lynn Margulis could evidence this. In the case of animal, in the case of eukaryotic cells, the eukaryotic cells engulf at one moment in the evolutionary times. For example, an alpha protobacteria, and in the case of chloroplast, probably cyanobacteria. Okay, so but despite she done a lot of experiments related precisely with mitochondria and chloroplast, because they have obviously they have a DNA genetic material. It's easier to find with an empirical view, with experiment, the similar characteristics between these two big groups. But now they are also exploring another kind of endosymbiosis, and another kinds of traits that it's possible to evolve, that have evolved from an endosymbiotic interaction. For example, in the case of nucleus, in the case of undulipodia, that is related to flagella and cilia in the cells. Or for example, this is in the case of unicellular organism, for example, or one cell, but it's also important for multicellular organism. For in this case, for example, Erythria chlorotica is a basal, it's a mollusk. sorry. So it's a mollusk that eats plants and conserve their chloroplast, and they put after their chloroplast in their tissues, in the external tissues, to make photosynthesis when they don't have food to eat. And for example, in the case of insects, it's very common to have endosymbiosis with different bacteria, as for example, the genera Wolbachia. And as we will see in the case of coral, it's very common this kind of endosymbiotic with a genera called Symbiodinium. And it's very common also in different, it's very profound and very wide in all the eukaryotic tree also. Okay, so that is one thing that is very important from an evolutionary view. In the case of the interaction point of view, it's also interesting because in the case, for example, in this case, I put in the picture a mitochondria to show that there is a very close relationship, interaction between the mitochondria and the nucleus of the whole cell. Okay, in the case, for example, of the mitochondria, that is an endosymbiotic organelle. Okay, mitochondria produce ATP or glucose, and then these products for the cells are relevant, are essential for their metabolism. And also the nucleus in this history, in this evolutionary history, produce a couple of reaction that also regulate the, for example, the reproduction of the mitochondria. Okay, so there is a relationship that is between the nucleus to the mitochondria and the mitochondria to the nucleus. And so obviously one big question that arises from these two viewpoints of endosymbiosis is how to study, obviously, an endosymbiosis as a complex process involving not only the interaction that I wanted to show there, but also their evolution. For example, how this interaction have changed during the million of years or thousands of years. Okay. I'm going to speak a little bit of this because we choose this coral and symbiodinium endosymbiosis to approach to this phenomena, to this general phenomena. Obviously, we have limits. We cannot describe. Hopefully, we want to describe different kind of endosymbiotic using one example, but this is something that obviously we need to develop more. But we choose this kind of endosymbiosis between corals and this unicellular organism, this alga, that is symbiodinium. In the case of this endosymbiotic is related to the corals that we can see here, that is part of, is a very early group of animals, and symbiodinium that is part of a chromal violata group. And in the case of corals, corals are a part of a big group that named Cnidaria, that is related to jellyfish, anemones, and corals. And for example, I wanted to, I am not sure if I can to do this. Okay, maybe it's possible. But we can see here a video of an anemone that I was eating. How this anemone eats. This kind of animals are very basal, very ancestral in this very group of animals. So I'm going to and symbiodinium, this unicellular organism live especially because here we have corals. Corals are very have a similar morphology to these anemones, for example, but they are very little. As we can see here, they are very little and have this kind of exoskeleton of calcium. Okay, and here in their tentacles, it's possible to find this unicellular organism, okay? Especially in their tentacles that we can see here, okay? And here we can, I am not, okay. Maybe the next one. Okay. So one important thing of this endosymbiotic is that the unicellular organism, the symbiodinium, that we can look here in green, lives in one of their layers, tissue layers of the coral. In the case of the coral, we know that it's very ancestral, it's very basic. So in the case of coral, and in general in this group, we have only 4 layers, okay? That is very basic in case of animals, okay? Because involves only 4 differentiation layers. One ectodermal layer, an oral endoderm, where the symbiodinium stay, And one interesting thing is that they have other 2 layers of tissue, but also an exoskeleton with calcium. And this endosymbiosis occurs in general, in a lot of corals, but and it's a mutualistic relationship in general. Corals need finally this endosymbiotic organism to live. And in case, for example, of starvation of the coral, this symbiodinium can give to the host the 50% of the energetic demands. Okay, so in the case of, for example, in some ecosystem that is not, there are not so much food, it's possible that this symbiodinium give a lot of food to the host. So, and from the other extreme, we can see that also this unicellular organism, this symbiodinium, can live inside the coral and benefits for their resources. So there is an interaction similar to the mitochondria with the nucleus. Okay, they can communicate each other by their resources and grow up as a consequence of this relationship. So one interesting thing that I want to note is that, for example, the host eats some resources that are in the environment, for example, phosphate or nitrogen, etc. And the symbiodinium use some of the products of the host, for example, the CO2, to promote more the generation of the skeleton. Okay, so this biochemical process that is promoted by the symbiodinium is also benefits to the skeleton and the organism in general, but also this skeleton promote also some reactions that benefits the growth of the symbiodinium. So it's a benefits that they benefit each other. And the other thing that is interesting also is that this relationship depends not only of this relationship, also is regulated by some host release factor. For example, if the population of symbiodiniums grow a lot, the host release factor begin to limit the growth of the population, and also occurs the opposite in using inhibitory factors also. So there is a regulation between the proliferation of the two organisms by this kind of signaling. So finally, in the case of this example, there is it's very interesting also because this relationship describes a relationship that have been persisted during evolution. For example, there are a lot of clades of a lot of lineages of symbiodinium that interact specifically with specifically hosts. So one interesting thing is to know what is the difference between those different interactions, and because they have a very profound evolutionary history, they also developed this interaction they also promote that this interaction begins to be mutualistic. What that means is that the host have a dependence of with the symbiodinium, and in this currently we know that there are a lot of corals that are losing because the rise of the temperature of the sea. They are losing their symbiodinium, this endosymbiotic relationship, and they die and begin to look white. Okay, so in the case of our model, we use we describe our model in general using this approach of this relationship of the symbiotic relationship that have different phases. For example, in the case of this relationship, we can describe an initial contact with the host cell and the symbiont, then a phase of engulfment, a sorting. Sometimes the host select one symbiodinium instead another, or something like that, and a proliferation phase, and another dynamic stability, and a dysfunction phase also. Okay, and we use specifically to describe, to approach to this model, only these two kind of phases: the proliferation phases, where that involves the regulation of the biomass of the host and the symbiont, and the dynamic stability phase, where typically we can find the metabolic exchange and nutrients traffic and the calcification of the host. And we didn't approach to the last phase. So we approach to this interaction using 3 levels. We describe a biochemical traffic level, a cellular proliferation level, and an organismic level. In the case of biochemical traffic level, involves the biochemical interrelationships, the nutrient traffics, for example, energy flows, and the production of the calcidodermis, that is the exoskeleton of the coral. And also the cellular proliferation level involves the cellular reproduction and death of the symbiodinium and the host cells. And the organismic level is the coral growth and protection against predators and other physical threats. And one other thing that we use to describe this interaction is some kind of signaling that in general is related. In our model, we relate this signaling to the level between the signaling between these two kind of levels, biochemical traffic and cellular proliferation level. So we use also, we need to have in mind that we use we have a layer, a metabolic layer, where the symbiodinium is living, and the calcium layer, that is the exoskeleton that so if we have these two layers in mind, I don't want to describe this reaction in profound. I want to only to mention and to show what kind of results we can have. But in this case, for example, we generate a set of species, the host cell resource, the energy, a resource to form the calcium layer, a resource that is product of the calcium formation reaction, a calcium, and we describe different reactions that are not too much, it's a very simple set of reaction that involves the inflow of the different resources of the system, the production of energy by the host cell, a calcium formation reaction, that this calcium formation is very important to promote this interaction, the potentiation of this interaction, an energy consumption, and an energy generation by the symbiodinium. Okay. And if you look, if you can look there, every reaction is promoted by a specific parameter. Okay. So after to generate, there were one image that I couldn't speak because I couldn't see I couldn't see in my slide before, but another thing that I forgot to mention is that after to have this set of species and these reactions, we can use this reaction to generate the specific differential equations, yeah, okay, as we can see there. And after that, we can see, we can compare different scenarios. For example, in this case, just an example, we can compare, for example, an intracellular dynamic, the biochemical level that we wanted to describe. Without endosymbiosis, we put, in this case, only one parameter in 0. And what are the results of the different species in this new scenario when the symbiodinium is present? And only to have these two kind of a scenario, we need to change only the parameter key 5. So in this case, we can see that the calcidermis and the energy of the system begin to grown up, and instead of this case, for example, where the calcium and the energy is very low. So we can describe a different scenario using these reaction networks. And also, in another publication that we have, we describe a biochemical level with this kind of reactions. If you look, this biochemical level is described because the reaction is reaction T1, reaction T2, etc. And we also describe a proliferation level reactions. Okay, so in the case of the proliferation level reaction, here we are describing, for example, the reproduction of the host cells, the reproduction of the symbiodinium cells, in this case. For example, the reproduction of both the host and the symbiodinium reproduce in this case, and the die of the species and natural degradation, for example, of other factors. In this case, for example, we use a specific, we use a factor that is a inhibitory factor to regulate the system. In this case, for example, we know that in the reaction P8, we have that when the host host cells and the symbiodinium cells produce the host as symbiodinium cells. Also, they produce an inhibitory factors that can regulate this equation, the KT7 that is before here. So we use these parameters to describe a kind of signaling between these two levels, the biochemical level and the proliferation level. Okay, I want to only just to show you this very briefly and very in an easy way. And as we can see here in this picture, for example, this regulation, I cannot show you very well, but this regulation involved, for example, we describe in that reaction, the host and the symbiodinium, that depends of that parameter key reproduction of the host and symbiodinium interaction, promote the reproduction of both. Okay, so that is one thing that we want to describe there. And this inhibitory factor, F factor, can affect the parameters of the reproduction of the host and symbiodinium, and indirectly the reproduction of the host and the reproduction of the symbiodinium. But also there is another parameter, because we looked before that there are host release parameters, but also inhibitory factors. So one thing that could happen is that when there is a lot of symbiodinium cells, there is an inhibitory factor that limits the growth of the population, but also there is factors that promote the growth of the population, and we named this kind of factor as an energy, because when the energy is very high, the population of symbiodinium begin to increase, but after gets a level of population, this stop to grow and begin to enter in this phase of stationary phase. Okay, so we wanted to describe here this kind of signaling that express this interaction between these two levels, and that is possible. We haven't described so well now, because it's obviously something that we are developing, we are working on, but it's also be possible to have an interaction, a signaling process between, or somehow an interaction between an organismic model and the proliferation level. Okay, and I show you only this picture. Okay, but we have in this paper, if you want to look at the paper, we describe the equation of that one. Okay, so for the final point that I want to address is that one interesting thing about evolution and development, or evolution in general, and one thing that I want to address is the application of the reaction network, and in specific the chemical organization theory, to evo-devo, to the development and evolution. How we can address the change of the form, the change during morphogenesis and during evolution. I think that this kind of approach could be very useful for this topic, for evo-devo, because in evo-devo, morphogenesis and evolution is a is a process, is a morphodynamic process that is very hierarchical. So we have a lot of levels here that interrupt each other, okay? And it's obviously we can describe one level, but there are some properties that we cannot only trying to research, trying to study only one level, because there are some emergent properties that arise when you study two or more levels. So and I wanted to use this picture because it's a picture of Waddington, very known, that show the epigenetic landscapes. And why I use this picture is because during development, obviously the different processes, different changes that occur during development are a consequence and also a promoters of evolution. Okay, in this case, in this picture, it's very abstract, but show briefly how one pulse can go for different pathways. And in these different pathways, depending of the state of the system, is how the ball can choose or can achieve one pathway or the other. So one interesting thing of development and evolution, I think that is modularity. Modularity obviously is modularity or discreteness is a universal emergent property. This is a definition of Mary Jane West Eberhard. Is a universal emergent property of organismic traits that results from the universal bounded responsiveness to inputs of the phenotype at all levels of organization. And continue, the bounded nature of a trait has to do with the bounded nature of its development as a product of a particular developmental pathway or a coordinated limited set of reaction or a localized developmental process. And I like so much this definition because she described that a modular trait then can be defined as a subunit of the phenotype determined by a decision point in development with a bounded endpoint, whether of morphology, physiology, or behavior. So I think that this image describes very well what happened during developmental process and how these models begin to construct during developmental process and how this model interact each other during the evolutionary process also. So in the case of modularity, obviously modularity has been studied for a long time, for decades, in evolutionary biology and in other fields actually, like neurobiology or different fields. But in general, have been studied using reaction networks. And for example, in these images, we show the modularity of the genotype and phenotype map. Okay, based, for example, in the case of circles, the genes, how they affect by the development some traits, and it's possible to describe in this review. They described how this could be used, but there are not so many tools to do that. How can be used to describe other levels of interactions, like for example, the genotype space or the mutational effect can affect the genotype space and also the developmental parameter space, the phenotype, and the fitness surface. some of the tools that are used, for example, in my field, in evolution, to describe this modularity in general, because we have a lot of level here. Obviously, we have the level of genes, the level of molecules, the level of tissues, the level of all the phenotypes and the organism phenotypes. And in general, for evolution have been used 3D geometric morphometric that use the form, the shape of an structure of an organism and describe the structure using landmarks and then use a statistical measure to relate the landmarks and find the models, but only using the shape, only using the external shape, the external phenotype. And another tool that have been developed the last during the last years is anatomical network analysis, where, for example, in this case, they show how some muscles interact more each other related to their structure and they interaction with bonds. Okay, so they can describe a modularity of the system, but based also in muscles and bonds. But it's also that also involves the structure and only these two levels that are very, very, very close related together. So one of the things that I wanted to speak a little bit is how this approach, the reaction network approach, could be useful to base it in the case that we have many species, many interactions, we can predict based in some mechanism their dynamic evolution, and we have analytics tools to study more this system, how we can, with this abstraction phase space, we can find probably using a big picture of the different levels, what are the organization, what are the subsystem that that can respond to this property of to be an organization. And obviously with this view of organization, we can relate this organization with a model, okay? But in this case, and I know that because Thomas told me that as a group, they are developing some softwares, some algorithms to generate a software to use a lot of species. I think that is possible not only in this kind of network, use structures, use species that responds to, I don't know, maybe muscles or bones, but also some species that are related to genes, that are related to molecules. And obviously, in the case of, because we have some parameters, some constant that we can move in different range, we can describe also, for example, the limits of I don't know how to say this, but the limits or how a molecule, for example, a morphogen can release and disperse in a morphospace, for example. So I think that this kind of tools could be very interesting to describe more and to understand more of how two models are constructed, are developed during embryology and during evolution. Okay, so I think that I'm finished. What? Okay. Yes. So I'm finished, and thank you for your attention. And thank you, Thomas, and thank you for everyone. I want to mention, obviously, Thomas, because we have developed this this work together. I put my email here, and thank you so much for all the institutions that support me always. Thank you.
Okay, so we have now available time for a round of questions. And here now we'll kind of delete this or reduce some stuff so we can start the discussion. If anybody has a question, please type it on the chat.
Yes, I think there are some in the chat.
Yeah, but no, they were like, hello.
Okay.
So I will bring the chat to this side, to this other side. So if the audience, the present, not see your screen, we can see now. Oh, yeah, but now that was fixed. So now in the attendance here, anybody has a question?
Well, the question is about the evo-devo. If I understood it well.
Sorry, Francis, before, let me check if everybody can listen to you. Can anybody confirm that Francis can be listened to?
Do you hear me when I speak like this?
We hear you.
Yes.
Good, thanks.
So my question was about the evo-devo. There is this famous model with the ball rolling into different valleys. Another way of understanding it is that a particular cell, when it is in the embryo, at a certain moment needs to make a choice whether it becomes, let's say, a liver cell or a muscle cell or a skin cell. It can do that by choosing one value. Another view would be that there is a dynamical system with a particular attractor. Is it then correct that what you suggest is that the organizations are then the attractors in which a particular group of cells may end up? Attractors of, in this case, an organization that means a number of genetical and other chemical reactions that are keeping the cell in a particular epigenomic state.
Okay, I am not sure if I understood well, totally well, but yes, I think that in relation to cells, when we are speaking from early stage of development, in general, we speak about tissues that are not totally differentiated. So when the cells begin, or when tissue begins to develop, they develop in a new space of different molecules, different morphogens. And in general, I think, yes, I think, as you said, that the cells change depending of this morphogen that in developmental process acts as attractors. So that is, and actually, I think that is.
I mean, my intuitive idea, if I understood it well, was that the organizations could be ways of formalizing the attractors, of saying this organization contains these and these molecules and these reactions. They keep each other, they are self-maintaining, and they define, let's say, the epigenomic state of a liver cell. In a liver cell, you need to have these and these things active, these and these genes active, and the others not. So it's a subset of all the possible molecules and reactions that could be there, but a subset that is self-maintaining, it is an organization. While in a muscle cell, there would be other molecules and genes active, therefore the reaction network would be a different one.
Yes, I am agree with you.
Can I join maybe to clarify? Because I think that the landscape, it's a sort of let's say, landscape of all pathways that can occur, and some pathways might end up in epigenetic configurations that do not keep going so easily, do not move forward so easily. So these are, I guess, somehow more stable cells, some more stable configurations of this. So
Yes.
I get the feeling that maybe we can think of this landscape like there are some valleys, and then you can remain somehow here. You are kind of resist epigenetic perturbations, and in other cases you are in like tipping points where whatever happens, you will evolve somewhere else. But I don't know if this is like exactly what you say. And then the pathways will be the ways.
I think the pathways are just, as it says, the ways towards the end state. But the end state is that either your liver cell or your muscle cell or your skin cell, these are really stable adapters, because it's very difficult to reprogram a muscle cell to again become a stem cell. We know that. So the pathways themselves are continuous, but the end states are discrete. There are discrete types of cells, and I suggest that each of these types of cells might be a particular organization of genes and molecules that are self-maintaining, but that are different from the other organizations.
Yes, actually, I understand you, and yes, I am totally agree with you. As Thomas said before, we have this, I like so much that picture, because the balls begin to be in a hill and is fall between different valleys, because one of those, because in this process, there is a differentiation of the cell, and it's very difficult, and obviously is something that is possible in an experimental in laboratory, for example, to change the difference, this cell that are differentiation to return to this initial state, because this initial state have more potential to have this to fall in this different pathway. But if one cell is in one pathway, the hills that are around that pathway, that valley, means that it's very difficult for this cell to put to another, to change to another differentiation. So I think that, yes, it's possible, and I try to show that in that picture with this segment A and this segment B, that because in this process of development, in general, not only cell, but also different, the cells, but the tissue also begins to change, begins to differentiate, but they are also bounded by constraint. And those constraints in developmental biology could be very different things. Constraints like forces of the tissue, for example, for example, other kind of tissues that are more dense, for example, and some morphogen couldn't diffuse to another more, couldn't more be diffusible or something like that. So
I wonder whether you could describe it in a Hasse diagram, that maybe the most undifferentiated state contains all the molecules, and then it goes down into more limited groups of molecules which then define the different differentiated cells.
You mean all the molecules like in a potential state? Yeah. I was thinking instead
or vice versa, it could be no molecules, and then it goes to a particular.
I thought in the opposite way, but maybe it's equivalent, that you can start from molecules that are highly reactive in many different ways, because that would be like the plasticity of the stem cells, I guess, that they can become anything. But once they change their state from being like very plastic to less plastic to less plastic, until you end up with a, let's say, a tree-like leaf, like the leaves of the tree will be the ultimate or the final configurations of the stem cells. But yeah,
that tree, I wonder whether that could be mapped onto
with a reaction network. Yes,
yes,
it would be a Hasse diagram actually,
yes.
and that's why the top of the Hasse diagram, the whole set of molecules, will not be an organization, because I mean, it could be an organization, but it won't be an organization reachable by a single pathway. It's like putting all the organisms together. So then you create first the cells, and then the collections of cells forming organizations. So that would be a new view on it. But my question at the biological level is why there is an ending process. Why do we have end states? Why stem cells change, develop, and at some point they become something that then biologists say, well, this is this. And why they don't continue, or do they, then their future is dying out? Or they just
think
continue?
That they always continue,
Yeah.
but obviously there are some constraints that are from our ancestry, from our evolutionary ancestry, that are very that are very limited. So obviously, but after that is because we speak not about developmental biology and not of embryology, because the development continues every time until our
because
death.
embryology assumes.
So in the case of it depends about a lot of things, obviously, but if you involve hormones or a systemic view, you know that this process, despite I think that you told what you told is very interesting, because when one cell or one tissue is organized and is limited, is with developmental constraint, it's difficult for one cell that is there change to another thing, but it's something that is always happening. And the cancer is an example of that, actually. But so this could be a good point also to understand those kind of diseases also.
Yeah, but let me kind of clarify something for myself. So the embryology is I'm putting this in perspective of timescales. So embryology would be a weeks to months timescale, right? Then developmental biology will be that until you die. So it will be like life cycle timescale, which might be like years.
Embryology refers to the development of organisms until they hatch. Or until they
each group defines its own time scale, but it's the shortest one. Then you go to
a little bit depends of the species, because
that's
there are some fishes that have a very huge developmental process, and then a life that is very very small, very short. Okay. And probably depends of a lot of things, but depends of the species. Yeah.
And the evolutionary time scale, it's anyways longer, because it has to be at least the sum of the two plus the heritage and all that.
It's possible to understand more about evolution using organisms that have a scale of development that is very, very, very short, as bacteria, for example. So there are some examples, for examples of evolution of multicellularity, or evolution of, I don't know, antibiotic resistance using bacteria or unicellular organisms, because they are shorter.
Theoretically, you can neglect the embryological phase, let's say. You can say, well, they just born complete. You don't have to incorporate the developmental process.
It depends. I mean, I think that you are referring that for some
doing theory.
Yeah, obviously.
It's easier to make a theory of an object that replicates the entire structure at once, and not something that it has a phase of growing and it has makes it more complex.
That is very important. Is something that is a thing that is possible to do, but one of my interests obviously is evolutivo, but
nice. More questions from the audience?
I wondered what is exactly the difference between like endosymbiosis and like multicellularity. So like when can you say we have two different organisms? When it's like one whole organism? Like
Yes, that is a very big controversy. Yes, because endosymbiosis is described and symbiosis. In symbiosis, you have two different species, two different organisms in general, and happen all the time in different lineages. For example, in the case of symbiosis, typical examples are lichens, but they are two big organisms. They are fungi and algae, but they describe as a symbiosis. But endosymbiosis is specifically when one unicellular single organism lives in the tissue of the other. So in general, it's understood in the symbiosis like that. But in the case of multicellularity, it's different because, in that way, in this kind of endosymbiotic system as coral and symbiodinium, it's a multicellular system. But the coral is an animal, it's a multicellular system. But one interesting thing is how this multicellularity arises. And that is a question that is very interesting for me also. If you want to, maybe we can discuss more about that. But because multicellularity at the beginning of animal arose because if you look how multicellularity develop in different lineages that are unicellular but are near to animals that are totally multicellular, they develop by aggregation of different cells. They are not clones, or by a reproduction of one cell that reproduce and begin to conform a unique organism, but with only clones. And then after a timing, their clones begin to differentiate each other in the multicellular system. But that's very, very interesting, yes. But no, are obviously when we speak of endosymbiosis, we speak about a multicellular system in general. I am not sure if
yeah, well, I guess it's a difficult question to answer. Like,
but
it's like, why is that one cellular organism not just part of the whole multicellular organism? Like, when is it like different organism and when is it like part of the same structure?
Like maybe bacteria that live on your skin or bacteria live in your intestines, are both of them considered endosymbiotic?
It's a little bit tricky because endosymbiosis now in general is related to a mutualistic relationship, and a mutualistic relationship is understood by ecologists that if you lose your endosymbiotic relationship, is something that is very very dangerous for your life. You can death, so you can die. So in the case of, for example, bacteria of our guts, it's not something that if we lose some population of bacteria, we obviously dysregulate the entire system. We can get an illness, for example, and that is something that generally happens with some illness, but we don't die. So I don't know, but yeah, I mean, it's a symbiotic relationship, yes.
That's not called endosymbiosis then, because
No.
Because you can survive the loss of your symbiosis.
Yeah. Because that kind of bacteria don't live inside the cells of that tissue. In the case of gut, they stay in the gut. That the gut is a whole that is like the environment, but in an internal environment. But it's not the tissue itself. It's not a cell that involve a bacteria, but there are other bacteria actually that that move between cells of the gut. Yes, like other, it's very interesting, but it's something that is possible, yes, to discuss.
Okay, we have a one question. Please open the mic. I think it will be here because the chat messages I was getting, they were sounding very loud. So probably you will be here. Please unmute. Hello. Good.
Do you talk to me, Thomas?
Yes.
Ah, okay. Wait, is that clear enough?
Yes, totally.
Ah, okay. Thank you very much for your very interesting talk. I've learned a lot of new things again. You mentioned decision making also as at some point. I was wondering how from the point of view of biology, you would classify where decision making really starts to take place. Do cells make decisions? Do bacteria make decisions? Do viruses make decisions? Well, humans make decisions, we don't doubt about that. Yeah? And what I particularly am interested, if you think about decision making in biology, do you think then about decision making as something where there are two alternatives, or whether, or eventually doubt is possible also, where there is no determinism, you see? It's just a question of how biologists think about that at the moment.
Yes, I am thinking how to how to approach to this question, and
perhaps I can add another thing. Yeah, how much it's possible to say, for example, that cells would collaborate to do a more complex thing inside of the body of an animal or a plant, where it would make sense to talk like that. I mean, scientifically, not just because of easiness of talking about it. Eh? Well, it's perhaps it's more a question of to know what biologists think about that kind of question at the moment.
Related to the decision making, what I think, and in general from a developmental viewpoint, it's not precisely a decision of a cell, because depends, that is for the reason that I mentioned before, this relation or this constant talking, this constant conversation between cells or between the organism, in this case a cell, between their environment, because the environment and the cells, the organism modify the environment, and also the environment modify the cell. So I think that is not a decision of a cell. I think that is a cell of the system, is a decision of the system. But what we have during developmental development is that this decision is, I think that is a decision of the system, is a conversation between these two factors, the organism and the environment, but it's something that happened obviously in a population. So just give me a second. But I wanted to say that is I think that is a decision between in the system, the the state of the system at the same point, at the same time. And obviously we speak about a morphospace, a space, a three-dimensional space. So all that also depends about the position of the cell in this space. I don't know if you understand. Did you understand?
Yes.
So I think that in general, the developmental viewpoint of the decision, in general, they think, or I think that is generally thinking, has generally thinking, that is that in general a cell is more passive to their environment. But I think that a cell obviously creates an environment also. So that is my viewpoint, but I am not.
Is it more or less the complexity you find in societies also, in human societies? Because you cannot say that the village will decide something. There's a council in the village and many complex interactions. Or is it still, can you interpret the interaction on the biological level in that way, or do you think that is not really a good way to think?
You could translate it to Spanish yourself. Dice que sí es como la misma idea que cuando uno le pide a pensar en que las decisiones las toma una ciudad. Las decisiones no las toma una ciudad, las toma, hay como una estructura interna que es la que implementa. Entonces, me pregunta si es la misma forma como de pensar en cómo toman decisiones las células.
I am not sure, I don't know so much about how a society takes decision, and I know that very not very deeply. But I think that could be the same, yes, because depends not only depends of the structure of the society. This depends of a lot of things. I don't know. Maybe, for example, the different cultures that are in the society, the different places that they habit, the context, the historical context, the historical context and the physical context where they are. So I think that obviously is something, I don't know that is the same, but it's a good analogy. I think it's a good analogy to approach to this problem. That it's possible to find a lot of analogies, yes. Yeah. I think so. But
let me perhaps tell you also why I ask this question, just to a biologist that you are, because the way that different decision structures appear can actually be an indication in which way, in which regime things happen, you see. In which regime, I know, but I've not studied this myself very carefully yet. But I know that Kranikov, you know Kranikov, Thomas, that he wrote some papers on decision-making of bacteries. But I should look myself more carefully into it. I don't know if it's very serious, actually, what he was doing there. And in decision making, there's so much variety more there than in just pure physical interaction, billiard ball like physical interaction, that by looking carefully to that, you can see much more the kind of regime that something's happening. That would be interesting in that sense, if biologists would pay attention to that, you see.
Yeah, yes. Actually, from an experimental view, it's possible to know, for example, how much gradients, how much concentration do you need for a molecule, for example, to generate a differentiation. So you can use this gradient to also, for example, for a reaction network, I think.
Yeah, probably. Another thing that inspired my question is, you know, perhaps that now in photosynthesis, which is interaction with light and living matter, they also have these models now with pathways. But for example, there they see that the pathways are not these nice landscapes. You really have much more complex landscapes, you see, for the photons who travel, the photons that travel through it, no?
Yes. Yes, that is the reason. Obviously, if we want to address more, to describe in more detail a biological system, is necessary not also to put the species and their interactions, but also to consider some kind of compartmentalizations or different things, because life or a cell, for example, is very compartmentalized. And if, for example, in your example, the photosynthesis occur for some proteins that are in some parts of the cell, exactly, in some parts of the chloroplast, some membranes specifically, and they need to stay there because the electrons move between that membrane to another place where there's another protein that is functional in that process. So yes, it's more difficult, but as another tools, for example, what I show related to 3D geometric morphometric, or in the case of anatomical networks, it's necessary always to do a reduction of the complexity of the system, for at least in the beginning, at least in the first models. So yes, obviously, I think that is the most ambitious idea that I have, is to also use this reaction network, but also put constraints to the system, morphological constraints. But maybe, I think, but I put this idea because I don't have in mind the model now, but I think that with the reaction network, because it's possible to use concepts, it's possible also to delimit, for example, some parameters to a diffusion. And with that diffusion, we can understand how they can change, but in a space. So I think that is maybe it's possible to do that. I am not sure. Yes.
Yeah, well, thank you very much.
We have another question, the last one, because of time. We are a little past the time. We started a little late too, so we're fine in that sense. But please, Christian, go ahead. Thank you.
Yes, Daniela, first of all, thank you very much for this rich talk, for this lecture. I have a question which maybe goes a little bit in the same direction like the question from Diederik. I'm intrigued by this pathway model. And when you said that there are certain decision points where the ball decides in which pathway it takes, I asked myself, is it this way? So does the ball decide, or are there forces playing on the ball that the ball takes this or that pathway? And so this is question number one. And question number two is, and I asked myself if maybe a pinball machine is a good image for this pathway model.
A pinball machine?
Pinball machine.
Pinball machine. Flip it.
Sí. Nice. First of for respect to the first question. Wow. Yes, I mean, it's something similar that I told to Dietrich. related to is not a decision of the ball. I think it's because maybe I am a little bit controversial now because I think that sometimes developmental biology think that is all the environment that make the decision for the cell or all the cell that make the decision for the environment, but it's the relationship between the both. So is it the system that make a decision in a specific context? So depends of the state. That is the idea that I have, and probably could be controversial to others, but yeah, it's in this picture. The ball sometimes obviously is an abstraction, but the balls could be in a hill and decide for one pathway or another, but that decision doesn't depend only of the ball, depends of the context. And some forces that you said depends of the forces of the constraint, depends of for example, some chemical constriction, for example, relation to morphogens that are proteins that can induce a change of the morphology of this differentiation of the cell, but also could be forces in the tissue. For example, there are some cells, there are some tissues that change, they differentiate depending of the force that they feel in that moment. And there are some proteins that are in the membrane of the cells that can translate or can transduce the environmental forces to the inside. So there is a talking that is between the external environment and the internal environment also of the cell. Is something that I cannot, obviously there are two systems there. There is an environment and there is a cell, okay? And the cell is an inner environment, but is probably an organization, but is in a constant talking with the external environment. So the decision is a consequence of both, I think. I am totally I am convinced of that, but convinced, yes, I am convinced of that, but probably it's a little bit controversial to others. But yeah, and respect to the second question, a pinball. So what I don't know. Depends of the pinball. Obviously, we can maybe design one because as Thomas said that I like so much also to do something related to robotics or something like that. So maybe you can design a pinball that represent this process. But I think that we need to get to understand what is the abstraction, because the pinball begins with you pull the machine that you
spring.
To spring the ball. So that is a force, that is something that you are putting to the system, but sometimes and probably every time in developmental biology is not totally like that. But yeah, have a lot of analogy also. I don't know. I need to think a little bit more about that.
Let me just clarify it again for myself now that we discussed. What is the ball? Is the phenotype or the genotype? What is the ball?
Depends. In the case in Waddington's picture, first of all, the first picture, because that is the first picture. They describe he described epigenetic landscape, thinking this ball as a cell. But this could be related also as an organism during evolution.
Yeah.
And also a group of cells during development. So it's an abstraction that could be related to a dynamic process.
Yeah,
You see the ball as the epigenome of the cell. The epigenome needs to decide which genes to switch on and which to switch off. That is, I think, what's the goal.
So then the pheno part is just assumed to be a direct consequence of the epigenome, which is also kind of doubtable. That was my problem. That if we assume that it's only the genetic part, then all the developmental part, the inactive developmental part is kind of neglected in the picture. Isn't
Not
it?
that actually he described an epigenetic landscape, and then he developed a two version of this picture, but putting the genes in a below in another level. So they put like a network of genes below, and then this hills and valleys picture to represent the plasticity of the expression of those
and
genes.
then the perturbations coming from the environment will be disturbances of this plasticity.
Yes.
And that will end up in a sort of affordance of what can be reached.
We are talking about a picture that was done. He did in the 60s, for example, in the 60s. So in that moment, obviously, this and he was an interdisciplinary profile. So profile. So it's not was very innovative in that moment. But now, obviously, we use this landscape picture to describe different things related to development. In general, they're related to development, but like developmental plasticity or
We should not forget that with the pinball, most big players they shake the cage also. They shake the machine.
Daniela, thank you very much.
Yes.
Thank you.
Any more question? Like nobody can take the last word, last thing to say?
Yes, maybe. If I might just add, because the pinball machine, I just made a joke about it in chat, but it introduces the idea of randomness or indeterminism into the play, because I think that's an aspect that somehow gets neglected sometimes in evolutionary theories, is that in order to have natural selection to be able to take place, you need a randomness or an indeterminism in mutations, be it internally or externally, to create the play field. Because if there's always forces acting from within or from externally on certain interactions taking place, then it's difficult to have a top-down process as natural selection taking place. I'm not sure if I'm making myself really clear, but I think it's important to keep that aspect of indeterminism in evolutionary modeling as well.
Yes.
As well as when I think about chemical organization theory.
I think that we can understand also our evolutionary background, our evolutionary ancestry as a constraint that promote also a direction. I don't know how to say that, but
about the pinball.
Sorry.
Yeah.
Okay, I'm sorry, but maybe if I think a little bit more, I can write you.
Yeah. Well.
Thank you.
Yeah. So I think we can start wrapping up and closing. Well, thank you, Daniela. Thank you, everyone, the attendance and the online attendance as well. and the future viewers of this video who are not here but watch it till the end. So with this, we finish our first seminar, and we will be planning the next ones very soon. So you will hear about this in the next future. So thank you very much, and well, feel free to write or propose speakers. We can organize also seminars in the future. Okay.
Thank
So
you.
thank you.
Great.
Bye.
Thank you very much.
Bye.
Bye-bye.
Thank you.
Bye. Thank you very much.
Okay. So
Now
now
you need to stop share. Or,
stop share.
yeah.
And now end.